Yeah, I'm Jack Hettema. Yes, I am now at Texas A&M, but I spent most of my career at Virginia Commonwealth University in the VIPBG Psychiatric Genetics Research Group under Ken Kentler. I have nothing to disclose. I'm actually a practicing psychiatrist. I specialize in the assessment and treatment of anxiety disorders. I've been studying their genetics for almost 20 years now. So it's a real pleasure of mine to talk to you about the progress we've made in the PGC Anxiety Disorders Working Group. Let me just remind you about the phenotype. Pathological anxiety, as defined in DSM-5, breaks down into social, specific, and agoraphobias, panic disorder, and generalized anxiety. These basically represent dysregulation of the normal human threat response, and the threat response is important in all species for the purposes of survival. But of course, like any bodily system, it can break down. Just like hypertension represents the dysregulation of normal blood pressure, anxiety disorders, we think, represent the dysregulation of normal fear responses. The thing about the human brain is it has such a complex set of threat responses that it can break in many ways, and thus these various manifestations of anxiety disorders. In fact, the phobias essentially represent excessive fear and avoidance to particular signals of acute threat. Panic disorder has all the manifestations of the fight or flight response, only in the absence of threat, similar to what Don Klein described as a false alarm. Generalized anxiety is more about future potential threat, where there's an excessive worry and concern and manifestations of anxiety. Our group doesn't investigate childhood disorders like separation anxiety disorder. And although PTSD and OCD are certainly anxiety-related disorders, they have prominent anxiety symptoms and used to be part of the anxiety section of DSM. Those are being covered by research in other PGC groups, so I won't cover them here. Anxiety disorders are extremely important group of mental disorders. I know every group says that, right? But they're very, very common. Probably the most common of different kinds of mental disorders. 1 in 7 lifetime prevalence, higher female to male risk in these. Their onset tends to be fairly early on in childhood or adolescence. And that onset then predicts future onsets of other anxiety disorders, mood disorders, and substance use disorders, which means by the time we study adults with anxiety, there's a lot of comorbidity. Their course is rather chronic, therefore they have a high burden of distress and impairment, and we've learned a lot about their genetics from twin studies. The heritability is only moderate, about 30 to 40% for the group, similar to major depression. And because of this high genetic comorbidity, it turns out genetic factors explain some of that, where there's a lot of shared genetic risk amongst the anxiety disorders, between anxiety and mood disorders, and with the predisposing factor of anxious personality, predominantly neuroticism and things like low social expression. So these are highly prevalent, moderately heritable disorders with a complex and heterogeneous symptomatology and high lifetime comorbidity. So they represent a real challenge to study, especially for genetic approaches. I want to introduce our PGC Anxiety Working Group, which joined the PGC in 2017. I share the chair position with Talia Ile from King's College London and Jürgen Deckert from Würzburg. We have a great team of analysts, Manuel, Kirsten, and Silviu. We have other people who have been supporting our work, Rosa Cheeseman doing a lot of the outreach, my colleague at Brad Verhoost, who's developing statistical methods to apply to these highly comorbid multivariate kinds of processes. And most importantly, I want to thank the 30 or more groups that have now been supplying us with data from their cohorts. We thank them very, very much for this great contribution that they make. And we've so far been funded by one National Institutes of Mental Health R01 grant. So this slide is my acknowledgement slide, by the way. But also our current analyses, we are planning or in the process of case control comparisons, primarily within European ancestry groups, like most of us start with. Our cases have a composite diagnosis of any lifetime anxiety disorder amongst the 5 I described before. Of course, we're going to look at individual anxiety disorders as the data allows and examine that expected and important overlap with depression and neuroticism. Future plans, of course, we want to expand to more diverse ancestry groups. There are some very interesting dimensional anxiety measures that function as endophenotypes. We want to collaborate with the other PGC groups to investigate the very important and interesting comorbidity with mood disorders, anxiety-related disorders, etc. And we are interested in the genetics of treatment response as well. So just to give you a little bit of history here, these are the most impactful anxiety disorder GWAS thus far. There are some other ones I don't have in the chart here, particularly out of the Tokyo University group, but these are the ones that contribute right now to the work we're currently doing. So there was a German panic disorder GWAS with about 900 cases in total. It was a 3-stage design. They did find association with a SNP in this TMEM132 gene. It was not quite genome-wise significant, but in subsequent analyses, there were independent replications. My group formed the ANGST consortium, and my postdoc Takeshi Otowa analyzed what at that time was a fairly large sample for anxiety GWAS, almost 18,000 people, only about 6,000 controls. So we didn't expect very much, but we did find one genome-wise significant locus in this non-coding long RNA. In 2019, there were 3 published papers, a lot of work done with the European Panic Consortium that collected almost a little more than 2,000 cases in their discovery cohort, had a large replication cohort, and had close to a significant association in this SMAD1 gene. Sandra Meyer, together with the eyesight group, analyzed anxiety and stress-related disorders. In other words, their cases had anxiety disorders as well as related disorders like post-traumatic stress disorder. They had almost 13,000 cases. They found one genome-wide significant hit in this phosphodiesterase 4B gene. Sandra Purvis from our group conducted a much larger analysis in the UK Biobank. The phenotypes are a little more limited with self-report professional diagnoses with some follow-up with the CD verification for GAD. They actually found 5 genome-wide significant associations. One of them, the most interesting, is in this NTRK2 gene, which is a receptor for BDNF. And most recently, Dan Levy, working with Murray Steen and Joel Galerntner in the Million Veterans program, looked at self-report professional diagnoses in almost 29,000 cases, and they found 2 genome-wide significant associations. So our work builds on these and actually combines data from these various sets with new ones. So we're still building our first sample freeze after 3 years. It's been a bit of a difficult fight along the way. With GDPR and COVID and all the other obstacles that many of you have also encountered. But we've conducted a preliminary GWAS meta-analysis with the samples that we felt were ready in terms of processing in QC. So they're listed here. I won't read them all, but some of the original ANGST data sets, some from the panic one GWAS by Forstner et al., the iPsych one, and then the UK Biobank GWAS, together with some ones from QIMR, Hunt, MOBA, ALISPAC, FinGen, Estonian Biobank, etc. Plus, we have a whole list of other ones that we're getting ready to add to this cohort to make a very large data freeze, we hope, of about 70,000 well phenotyped cases, and a total of about 130,000 cases in total with 750,000 controls. But for right now, our preliminary analyses apply to 68,000 cases, 460,000 controls. And again, it's any case, any anxiety disorder case, and what we call affective controls, where the controls have no anxiety or depressive disorders, given the very strong genetic overlap between these syndromes. Okay, so here's our preliminary meta-analysis. We found 12 genome-wide significant regions. The QQ plot is as expected for a polygenic disorder with a very respectable lambda 1000. Here's a list of those 12 associated regions. Don't pay too much attention. They're likely to change in the next few months as we add more data sets and conduct more comprehensive analyses. But I just point out that with these starred regions here, here, and here, these also were reported in the Ray et al. PGC major depression GWAS that came out just a few years ago. So here's some of the expected overlap with genetic risk for major depression. Also, there's this SORX3 gene that was not only found previously associated with major depression, but also ADHD and other disorders as verified by the cross disorder analysis as a highly cross disorder relevant gene. We conducted some internal replications with the major data sets via leave one out analyses, and we are reassured that there's high replicability here with these close to one genetic cross correlations. Here's the usual expected plot of cross phenotype genetic correlation, high correlation with things like PTSD, neuroticism, and depression, more intermediate correlations with things like bipolar, autism, and schizophrenia. We went ahead and peeked a little bit at the functional analyses using FUMA, and the gene ontology databases came up with the expected or hoped for kinds of biological pathways having to do with synapses, particularly pre and post synaptic membranes. We have neurotransmitter vesicles, cation neurotransmission activity, et cetera, et cetera. So that's, again, all reassuring. Also reassuring is the preliminary tissue expression analyses we conducted for these association signals, and they entirely occur, as far as we can tell, in the brain. And then these are probably a little more preliminary, but we looked at the cell type specific data that's available using RNA-seq in specific cell types. And very interestingly, GABAergic neurons showed up as the primary tissue, which is somewhat surprising but reassuring to me, because we've known for a long time that benzodiazepines work well to treat patients with anxiety disorders through GABAergic neurotransmission. So very preliminary, we'll see if it holds up. So where do we stand now in terms of the newest kid on the block having joined the PGC? This is from the now already outdated figure 3 from the Sullivan and Geschwin Cell article with the numbers of cases and genome-wide significant loci from the various PGC groups. This is that famous plot where there's a point here where the number of genome-wide significant loci that are discovered based upon sample size turns around and becomes linear. Well, for the more highly heritable disorders like schizophrenia, bipolar, and ADHD, this occurs at a lower number of cases, somewhere between 20 and 30,000. For major depression, it occurred somewhere around the 100,000 case mark or a little higher. And this is where we sit with our preliminary analysis with about 68,000 cases. So we think we're somewhere in between, as our heritability estimates would suggest, and we'll be very interested to see where this takes off when we add the other cases to our freeze. So after 3 long years, we feel like our group is finally starting to rock and roll. Unlike Stefan Ripke a number of years ago at this conference, I can't slide out on stage playing the electric guitar, but those who know me know that I've for a long time been a drummer in classic rock band. So we're really, really happy to share these preliminary results with you. Tomorrow at PGC Day, Jürgen, Talia, and I are holding a virtual coffee and tea break, so join us if you want to talk more. Thank you very much.